Austin's going to be coming around and passing you out a piece of paper that's an extra credit opportunity. And you will find the answers to the questions in the lecture today. So if you stay awake and you pay attention, it should be a freebie for you. Yes, everybody text all your friends really quick. Dude, get out of bed. Come on. We're going to pick up with the topic of cell membrane structure, which we didn't get to do because of the weather emergency before. And with any luck, we'll get through that, move on to membrane transport, and then we'll actually be caught up. We'll be right back where we were supposed to be. So you find lots of membranes in cells. If you're a prokaryotic cell, it's pretty simple. For a lot of them, you pretty much have one. That is the plasma membrane that surrounds the cytosol. Some types of prokaryotic cells have an outer membrane as well as a plasma membrane, and then there's some photosynthetic ones that have some interior membranes, although they're not exactly the same as the closed compartments that we find in eukaryotes. But in a eukaryotic cell, you've got both the plasma membrane and then all these internal membranes, and those include things like transport vesicle membranes, organelles like the Golgi apparatus, the mitochondrion, the lysosome, the peroxisome, the nuclear membranes, the endoplasmic reticulum. All of these different membranes have the same basic structure. However, the compositions of those structures are not always the same. Now, the plasma membrane has some important functions in any cell, and there are 4 of them summarized here. First of all, it's a barrier. And so its job is to restrict what can get into the cell, what can get out of the cell. And this is going to be inherent to the structure of this plasma membrane and to all other membranes as well. That is, things don't just easily move across them. There needs to be a specific system in order to do that. Plasma membrane is also important for receiving information. The signals that cells get from their environment have to get across this membrane in the interior of the cell so they can be interpreted. The import and export of molecules to the cell intimately involves the plasma membrane. And so there are going to be specific systems that will determine whether a particular type of solute, can get into a cell or not, or if it can get out of a cell or not. Finally, the plasma membrane is key to something that cells do from a mechanical point of view, and that is to change shape and to move. And so the capacity for actual expansion, contraction, movement is one of the most important functions of the plasma membrane in any cell. The plasma membrane and all membranes have as their backbone structure a bilayer of phospholipids. So when I say bilayer, what do I mean? I'm asking about the word bilayer. What does that mean? There's 2 of them. There's 2 layers. And we call the individual layers monolayers or leaflets. And there is a pair of leaflets that are oriented toward one another such that the nonpolar tails of the lipids that make up this bilayer are facing toward one another and away from the water environment on either side of the membrane. This is the plasma membrane of a cell, so out here we would have the external environment, and here we'd have the cytosol. And facing those water-based environments, we have polar head groups of lipids here. All of the lipids that make up membranes are amphipathic, meaning that they have both polar and nonpolar parts to them. And so you might typically diagram it in this way here. Here are some long hydrocarbon tails representing the nonpolar regions of one of these membrane lipids. Here is a hydrophilic or polar head group of some sort, and there are all kinds of different head groups. And these are what's going to be facing outward toward the water-filled environment. There are many proteins that are associated with membranes. We'll be taking a closer look at those. And then there are sugars, and the sugars can either be attached to proteins, shown here, or they can be attached directly to lipids. A protein with sugar attached is called a glycoprotein. Lipid with sugars attached is called a glycolipid. And you find lots of glycoproteins and glycolipids in membranes. Looking at the cartoon there, what's something interesting that you notice about where the sugars are? Well, there's some on the protein, there's some on the lipids, but what else do you notice in that diagram? And it's not just random, it's it's real. They're only on one side. They're only on one side of the membrane. We'll talk about why that's true. Oh, I missed one little thing, and I'm just saying it not because you need to remember it, because it's interesting. Lipid bilayers are pretty much universal to cellular membranes with one exception, and that is in the weird group of prokaryotic cells that we classify as the archaea. And many of the archaea have completely different type of membrane structure. Rather than having a bilayer of amphipathic lipids, they have a monolayer of a type of so-called ether lipid in which you've got something that looks kind of like an Oreo sandwich cookie. You've got a hydrophobic or non-polar portion to each ether lipid molecule, and then you've got polar head groups on either end of it. So instead of a non-polar and a polar end, you got a polar, non-polar, polar, all in one lipid, and these things will form a monolayer. The reason for the existence of those structures is many of the archaea live in environments that are rather extreme from a chemical or thermal viewpoint. It's either very, very hot or very nasty and acidic or salty. And a lipid bilayer membrane, which is held together by forces that are non-covalent, simply could not persist. It would fall apart. And so the archaea had to come up with something special. We're not going to look at that anymore. You don't have to remember that. But I think it's an interesting thing to know that even though we tend to treat this sort of structure as universal, it's not entirely so. Okay, the major groups of membrane lipids are illustrated here. And those are going to include the phospholipids, the sterols, and the sphingolipids. One big group of lipids you don't see here that we talked about before when we were doing molecules is the triglycerides. Anybody remember the triglycerides or triacylglycerols? Those are your fats and your oils. You don't find them in biological membranes because they're not amphipathic. They're completely nonpolar, completely water insoluble, and they wouldn't function well in a structure in which part of the membrane needs to be exposed to a water environment. So you're not going to find your triacylglycerols here. The phospholipids have a common structure. Their backbone is the 3-carbon alcohol glycerol. Glycerol has 3 hydroxyl groups on it. 2 of them become esterified to hydrocarbon chains, and those are very nonpolar. And the lengths and the degree of saturation in those hydrocarbon chains can vary. The 3rd hydroxyl group forms a bond to a phosphoryl functional group, PO4. That's a negatively charged group, so there's some polarity right there. And then the other side of the phosphoryl group is going to become linked to some kind of other polar head group. And there are lots of different kinds of polar head groups. Some of the major ones are choline, which is shown here. It's a nitrogen-containing compound that has a positive charge. The amino acid serine is shown here. Serine is a zwitterion, I said earlier, at the conditions that exist in cells, so it has both positive and negative charges, which tend to kind of cancel each other out, but they're still polar. Then there are other amines like ethanolamine, there are sugars. All kinds of polar groups can be attached here to give you a complete phospholipid. And they have different names: phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine. The phosphatidyl part is the key. And then what's tacked onto that tells you what the head group is. The sterols are a very different kind of lipid. They have a set of 4 almost planar rings that are joined to one another. And then there is a short hydrocarbon tail attached to the 4th ring. But on this first ring here, there's a hydroxyl group. Hydroxyl group going to be polar or nonpolar? It is polar because oxygen is very electronegative, hydrogen is not. And so if you slip this into a membrane, which side do you think would be facing toward the water? Yeah, the polar, the hydroxyl end. So these things are going to fit alongside the phospholipids so that their hydroxyl groups are lined up with the polar head groups of the phospholipids. Then the sphingolipids are the last major group. They're an interesting bunch, very diverse. They got their name, sphingo, from the Sphinx in Egypt. When they were discovered in the 1800s, the Sphinx was a great fascination for folks in Europe. And they used it as sort of a symbol of anything mysterious. And the chemist who discovered sphingolipids was a guy named J.W. Tüdeckem in Germany. He found them very mysterious, and so he gave them this name sphingolipid because they were so weird, so different from the other lipids that he and other chemists were studying. A sphingolipid has a couple of hydrocarbon tails, but one of them is not from a fatty acid, or not directly from a fatty acid. It's actually a compound called sphingosine, and sphingosine is formed by taking a fatty acid and joining it to the amino acid serine. And then you couple that to another fatty acid here, and then finally you add a head group of some kind here. In this particular sphingolipid, which is called a galactocerebroside, you add simply the sugar galactose here. Notice there's no phosphate group in this one. Okay, this is a neutral sphingolipid. There are other sphingolipids that do have a phosphate, just like phosphatidylcholine and the other phospholipids do, and it would be stuck in right here. Sphingomyelins are the group of sphingolipids that are phosphorylated. And so they, like phosphatidylcholine and the other phospholipids that we talked about earlier, are going to have a very polar head group, quite often have negative charge because of that phosphate group. Okay, why do these membranes exist in the first place? And I'll give you a cheat right up front. It's not because some force in the cell puts them together. It's not because there are enzymes that assemble them into this bilayer. They self-assemble into this sort of a structure. And the question is, why does that happen? Well, it all really goes back to thermodynamics. So picture a water-filled container, and into that container you drop one poor lone little phospholipid molecule. So it's bobbing around in this sea of water. Water molecules are all forming hydrogen bonds with one another, and they can form them in all kinds of different ways. They can form them, break them, they can twist, turn, everywhere they look. They can form a hydrogen bond until they run into our phospholipid. Now, they can form hydrogen bonds with the head group, but what about the nonpolar tail? What happens there? No. This is a common misconception. There is no repulsion. Stop and think about a water molecule, right? It's got partial positive charges and partial negative charges on it, right? And negative repels negative, positive repels positive. Are there any partial positive and negative charges in this hydrocarbon tail? No, no. The bonds are all nonpolar. There is no unequal distribution of charge, so there's nothing to repel. So water doesn't repel the hydrocarbon tails. It just simply can't do anything with it. It can't interact with it. So any water molecules that face this hydrocarbon tail got no hydrogen bonding partner. Therefore, if you totaled up all the number of hydrogen bonds that you could form in our container of water, before and after you put this phospholipid molecule in here, there's less now that the phospholipid molecule is there. And in fact, the water molecules form kind of an ordered cage around it, where they make as many hydrogen bond contacts with each other as they can, but of course nothing happens with that hydrocarbon tail. That means, believe it or not, that the entropy in our container of water with our one lone phospholipid molecule in it is slightly lower than it was with just the water, meaning it's thermodynamically unfavorable. The water would like a way to form more hydrogen bonds. Well, let's put two more phospholipid molecules into our environment here. Everybody is moving around with thermal energy. They're all pushing and pulling. They're moving randomly, and eventually three of our phospholipids encounter each other. Now, their hydrocarbon tails are not going to interact with each other from a distance. They're not going to be pulled toward each other, but the water molecules are pushing them around. And if the water molecules happen to push them together, and you look at what's the total number of hydrogen bonds I could get if these guys are like this, versus if I had three of these floating around in different places, you'd actually have more here than you would there, meaning the entropy is just a little tweak higher. And if we took this kind of a structure and now took two copies of it and turned them such that these hydrocarbon tails were oriented toward one another, you'd have more total hydrogen bonds here than you would have if six of these guys were floating around individually. And so oil and water tend to separate. Polar and nonpolar tend to segregate because of random motion, not because of any sort of repulsion. But once you get the nonpolar stuff together, you actually increase the number of hydrogen bonding partners for water in the mixture. Therefore, water's not going to want to go back. It's not going to want to push this structure apart and go back to a situation where you had six randomly distributed phospholipid molecules. And so in the end, you can think of what we call the hydrophobic effect, the tendency of nonpolar and polar molecules to separate from one another as kind of an entropy situation. Entropy is highest when you keep these guys with others that are like them and allow the water to interact with other water molecules or with other polar solutes that are present. And so that's why oil and water separate. That's why phospholipids tend to form structures like bilayers when they're in a water environment. Now, the bilayer is not the only structure that phospholipids can form. They can form things like micelles, where all of the hydrocarbon tails turn toward each other and form something like a sphere. They can form vesicles, where you have a bilayer that encloses a region that is water-filled. A vesicle is just a microcosm, if you will, of a cell. That's basically what a cell is. Yes? It does if you have those things separated from each other. What you got to do is compare, what if I had 20,000 in a mixture with water, and I somehow made them all stay apart from each other, and I measured the total number of hydrogen bonds there, versus I allowed water to push these things around until they got crowded together. And then what's the total number of hydrogen bonds there? It's higher in that second case, because what you're doing is you're essentially taking a large part of your volume and saying that's hydrocarbon land, that's nonpolar land, and we, the water molecules, don't have to deal with that over there. Okay? So again, it's really more about entropy than anything else, but maybe an easier way to think about it is just how many different ways can water twist and turn and make hydrogen bonds. And it's going to be higher if the water phase is separate from the nonpolar phase. And so that's why membranes tend to form spontaneously. That's why vesicles tend to form spontaneously. And you can do this yourself in a lab. You can just throw some lipid in water, and you can actually cause this to happen. And people do it all the time. They make artificial membranes so they can study the properties of lipid bilayers. Okay, the lipid bilayer is not a rigid structure because the phospholipids are not covalently linked to each other. They are held together by a kind of force. Anybody think of which one it would be? Think back to all our non-covalent forces. Which one would make sense? Somebody said something. What was it? Van der Waals. Very good. When you're crowding together all of these nonpolar molecules, they actually get close enough to each other that the van der Waals effect becomes significant. That is, individual atoms are creating transient dipoles with one another where you keep attracting, repulsing, attracting, repulsing. But the net effect is to tend to keep them together once the structure has formed, so they don't want to disperse again. However, lipids can move within a bilayer. They can flex back and forth, particularly the hydrocarbon tails. They have a lot of freedom of motion there. You have a lot of carbon-carbon single bonds. They can rotate. They can spin in place. And they can diffuse laterally or translate within one of the monolayers of the bilayer. So they can move this way, they can move that way, they can move this way. The one thing they don't tend to do is to flip-flop, to cross from one side of the bilayer to the other, to go from one monolayer to the other monolayer. Why would that be true? Why wouldn't that happen very often? What has to pass through what? Okay, something polar and something nonpolar are in conflict. What's the polar thing? The head group, okay? And what's the nonpolar place it would have to go through to get to the other side? The interior, where all the hydrocarbon tails are. And if membrane absorbs some thermal energy, and it's doing that all the time. And remember, thermal energy doesn't mean it has to be really hot. We've got plenty of thermal energy in here, even though we can be quite comfortable. But an individual molecule can get enough thermal energy, and maybe it starts to want to go this way. But when it does that, what happens? All the water buddies over here are going, hey, come back. We were hydrogen bonding. We were having so much fun. Come back. And so it tends to do that. It tends to pop back. So it would have to absorb quite a bit of kinetic energy in order to actually flip-flop spontaneously. That does happen at a measurable rate, but it's really, really low. Doesn't happen very often. However, that doesn't mean that flip-flop doesn't occur. It does. And when it does occur, it's mediated by some of those proteins that are in the membrane. Now, what are the sterols doing in the membrane? Well, actually quite a bit, as it turns out. If you look at a sterol schematically, you've got this sort of rigid planar part where the rings are. You've got the polar hydroxyl group up here, and then you've got this fairly flexible nonpolar hydrocarbon tail. So if you slip a sterol in between a couple of phospholipid molecules, it's going to orient this way, like we said before. So this portion of the hydrocarbon tails of the phospholipids is going to be seeing or interacting with the nonpolar ring structure of the sterol. And that's fine. They get along fine. They're both nonpolar, no attraction, no repulsion. Water doesn't want to come in and live there. And then the hydroxyl group, it gets along fine with the polar head group here. This guy, he's down here with the most flexible part of the tail. So sterols get along just fine with phospholipids. But it's this rigid ring structure here that explains why sterols are important in membrane. They actually restrict some of those types of motion that the phospholipids can have. The flexion, the spinning, the translation, the diffusion from side to side. Sterols restrict that. Therefore, a membrane that contains sterols is actually less fluid, or another way to think about it is more rigid than one that doesn't contain sterols. And that's important because at the temperatures at which many living things exist, if you had a pure phospholipid membrane, one with no sterols at all, it would actually tend to come apart because there's not enough collective van der Waals forces and hydrophobic effect from the water molecules to overcome all that thermal energy. And so the membrane would be kind of like sloppy jello. It would tend to come apart, and that's no good if you're a cell. You do not want your membrane losing its components and becoming leaky. So the sterols tend to make the membrane more rigid and less permeable. They keep it from coming apart, even when the temperature gets up in the 90s or above. However, perversely, if our organism goes someplace where it's cold, where there's a lot less thermal energy, a membrane without sterols would have a real problem because you've got all these hydrocarbon tails that are mostly unsaturated, a lot of freedom of movement, a lot of ability to rotate around those single bonds. That means that you can pack those suckers together really tight. And when that happens, the membrane loses all fluidity. Nothing can diffuse around. It's kind of like when bacon grease sets up in the pan when you leave it sitting around. And that's not going to work for the cell either. But these same sterols that made the membrane more rigid and less apt to come apart at warm temperatures now keep those phospholipids from packing too close together because of that rigid four-ring structure there. And so the membrane is actually more fluid than it would be in the absence of the sterols. Now, that doesn't mean it's more fluid than it is at warmer temperatures. It's not. But it's more fluid than a sterol-less membrane would be. So the sterols in our membranes are critical to their function. They could not do what cells need them to do if they didn't have those sterols there. And that brings up an important point. We all think of cholesterol as poison, right? We have been trained to think of it as evil, just like we're now being told that gluten is evil. You know, anything with gluten is bad. You know, you need to go dump it in Lake Michigan. Gluten is bad for certain people. Do you know who it is? Anybody in here have celiac? Yeah, if you've got celiac disease, you should not eat gluten. It will make you very, very sick. If you don't have celiac disease, eat away. Gluten is a protein. It's what makes your pizza dough stretchy, okay? And most of us can digest gluten just fine. And so all of this fad now about gluten is a toxin, get it out of your diet altogether. You can do it if you want to, and if it makes you feel better psychologically, that's fine. But unless you've been diagnosed with celiac, nah, it's not really doing anything for you. And the same kind of goes for cholesterol. Our bodies make cholesterol. They make quite a bit of it. It's made in cells. In multicellular organisms, it's made in one particular organ. Anybody know which one it is? Which organ in your body is the main site of cholesterol biosynthesis? No, that's not an organ. It's an organ L. I heard somebody say it. The liver, that's right. The liver controls your cholesterol supply, and it doles it out to the body through the bloodstream. And so your body is great at making its own cholesterol, and it's great at controlling the amount of cholesterol that's available to all the cells. And it doesn't need any help from you. The problem comes when you eat a lot of animal fat, which has a lot of cholesterol in it because the animal cell membranes had cholesterol in them. Now you're taking in more than you need. And as long as you don't take more than your body can handle, everything's okay. But when there's more than your body knows what to do with, it starts trying to find things to do with the cholesterol. And what they end up doing is getting packaged into particles that get deposited on the linings of your blood vessels, and you end up with atherosclerosis and heart disease and all kinds of wonderful things. So the evil of cholesterol is really more about our behavior than it is about cholesterol itself. It's about the way that we eat. And as long as you match your intake to what your body can handle, you'll be all right. And you could not, in fact, live without cholesterol. You know, some people say, oh, aha, I'm just not going to eat meat altogether. That's fine. I won't get any of that evil cholesterol. If you eat plants, you're eating something called a phytosterol. Here's one right here. It's called beta-cytosterol. Look at the structure compared to cholesterol. Right across the board. Only difference is right here. Okay? If you like to eat mushrooms, you're eating ergosterol, fungal steroid. Very similar again. Differences down here in the hydrocarbon tail. Now, it's true, our bodies can't process these guys. And in fact, they inhibit some of the enzymes that are involved in cholesterol biosynthesis. So it's actually not a bad thing to eat these food products that contain other sterols than cholesterol. But there's no there's no plant product that contains cholesterol. And this gets to another marketing ploy when you go in the grocery store and there'll be all kinds of things on the shelves that say cholesterol-free, cholesterol-free, cholesterol-free. And you say, thank God, General Mills, you took the cholesterol out for me, so I'm going to be healthy. If you look at all of those things, what are they? They're all plant products. They're all plant products. So they don't have any cholesterol in them because there was never any cholesterol in the raw materials to begin with. So all the company did was put a label on the box and count the cash. Okay. In prokaryotic cells, we don't find sterols at all. We don't find molecules that have that sterol nucleus, shown right here in green. But you do find something called a hopanoid. That's shown here. And from a functional point of view, you're looking at something awfully similar. Rigid multi-ring structure with some hydrocarbon functional groups here, methyl groups. And then you got one lone hydroxyl group up there, polar end. And these appear to do exactly the same thing for bacterial and some archaeal membranes as the sterols do for eukaryotic cells. That is, they help to modulate the fluidity of the membrane. They help to keep it from being too loose when the temperature is high, too rigid when the temperature is low. So sterols and now hopanoids are critical to membrane function. Said that lipids can diffuse around in a membrane within the plane of their bilayer and only very rarely flip-flop from one side of the bilayer to the other. Proteins can move too. And that was demonstrated conclusively in an experiment done in 1970 by two people named Fry and Edidin. And what they did was they took advantage of some new technology that became available in their day. One of them was the ability to get cells to fuse with each other in a laboratory dish. As you could take a cell from one tissue and another tissue and you get them to fuse together and make a hybrid cell. Or you could take cells from different animals and get them to fuse together. The other technology was the ability to take antibody molecules, immune system proteins that are ligand specific. Each antibody binds one particular molecular shape called its antigen. And then in a test tube to join to that natural protein a fluorescent chemical group. So wherever the antibody went, you could track it because you could see where the fluorescence was. So what Fry and Edidin did was this. They took human cells and mouse cells, and the mouse cells had on their surface an antigen called their H2 antigen. It's part of their major histocompatibility complex. H2 antigen is not found in human cells. They have their own major histocompatibility proteins. There was an antibody that would recognize the mouse H2 protein and another antibody that would recognize the human protein, but neither antibody would recognize the other species protein. Then they coupled a green fluorescent molecule to one of the antibodies and a red fluorescent molecule to the other one. They took a mouse cell and a human cell, and they fused them together and then immediately dropped the temperature, made everything cold. And then they added their two antibodies. And what they saw was that you would get green fluorescence on one side, red fluorescence on the other side. That is, you would have this cell you could see under the fluorescent microscope that was like a basketball cut in two, half green and half red. So you had mouse proteins on one side, human proteins on the other. Then they raised the temperature up to normal body temperature for a human anyway, not a mouse, 37 degrees Celsius. And they just let it percolate for a while. Then They cooled the cells off again, and they did the antibody experiment again. And what they found now was a random distribution of the fluorescence of the two antibodies. Only possible interpretation of that is that the proteins had moved during the time that the temperature was raised to normal body temperature. And since that time, many, many other experiments have been done that show that, in fact, proteins diffuse laterally in membranes, just like lipids do. So everything has the potential to move. And that has led to more work that shot down a very early idea about membranes, which is that they were kind of homogeneous. That is, that because there was this fluidity, because things could diffuse, that in fact the proteins, the lipids would all be randomly scattered in this membrane at any one time. We now know absolutely that's not true, and it mainly comes from work with fluorescent antibodies, kind of like the experiments that Fry and Nedden did. And so this is just a figure out of a paper that was done in Frontiers of Plant Science, and it shows the localization of different proteins or lipids here as measured by the binding of fluorescent antibodies. And so you can see the clustering of this green here, the red here. In these particular transformations of the fluorescence activity, you can see tight clusters of particular molecules. And we know that that's true now. So membranes are absolutely not homogeneous, even though things can diffuse in them. So why is that true? Why do some things cluster together? Well, one proposal that became instantly popular and is still very popular in a lot of the scientific community is that there are things called lipid rafts. What a lipid raft is it's an association of certain types of membrane lipids, specifically glycosphingolipids and sterols, with not just any old random protein, but certain proteins, certain important proteins, and that these things cluster together and they form their own little phase within the membrane, and these things diffuse around. And you can find all kinds of animations on YouTube that show a conception of what this might look like. As I said, lipid raft hypothesis is very, very popular, and there's lots of indirect evidence for it. Problem is there's no direct evidence for it. Nobody's ever seen one. Furthermore, some of the evidence that first led to the lipid raft hypothesis has been shown to be an artifact. Anybody know what an artifact is in science? Okay, you're thinking of in anthropology or archaeology what an artifact is. This actually isn't even spelled the same way all the time. A lot of times it's spelled with an E instead of an I. What's an artifact in biology or other sciences? No, it's not like a theory. Anybody ever heard the term used? Yeah, that's an artifact. That's artifactual. What? I'm sorry, what? You're getting toward it. It's not just random. Yes? Okay, a distortion. Yes? Okay, it doesn't have actual basis. That's probably the closest to it. That is, it seems to show something, but it's not. It's a result of something unrelated to what you were trying to study. Okay? And artifacts happen all the time. And in fact, papers get published and then withdrawn later when it's shown to be an artifact. That is, oh, this isn't something that's really in the cell. It's about the way that we treated the cell when we were trying to take it apart. And that's what caused this to happen. And it's a real bugaboo for scientists. You have to be very, very careful. That's why lots and lots of different people have to look at the same thing in different ways before you decide, yeah, that's really happening in the cell. Because if somebody's doing the experiment a particular way, they can cause something to happen unknowingly that would result in misinterpretation of the results. Years ago, I read a satirical book called The Book of the Subgenius, and in it the author advocated that we all walk around with the most random junk in our pockets that we could think about, so that after civilization dies and future archaeologists come back, they'll be totally flummoxed by what we were doing. That would be a good example of an artifact, that is something that isn't what you think it is. Okay, where do membranes come from? Well, I mean, the simplest way to understand it is that because cells only come from other cells, that is, you can't make a cell just out of organic and inorganic chemicals on the modern Earth, that every new cell inherits membranes, and that's absolutely true. So you don't have to completely synthesize a new plasma membrane. You don't have to synthesize a new endoplasmic reticulum or a nuclear envelope. But in fact, you do have to make these things because membranes grow, they expand. Cells need to package things up in vesicles. They need new membrane for that. And so membrane synthesis does occur in cells. And where it occurs is in part of the endoplasmic reticulum. Anybody know which part? Smooth or rough? The smooth. Yeah, in the smooth ER, that's where you find the enzymes that are involved in the synthesis of membrane lipids. And specifically, you find them on the side of the ER that's facing the cytosol. So you can think of that as the outside. So remember, the ER is like a tube in cross-section like this. The part in the middle, the cavity in the middle, is called the lumen, or the non-cytosolic portion of the ER. And then the part on the other side of the membrane, that's the cytosolic part. So the enzymes involved in lipid biosynthesis for membranes are found on that cytosolic part. Some of them are actually in the cytosol themselves, enzymes that couple fatty acids onto glycerol are found there in the cytosol. And the fatty acids have to be carried around by proteins that they stick to because they can't just float around in a water medium. And so those proteins deliver the fatty acids right to the surface of the ER, and there these membrane enzymes are that are going to link them to glycerol to create diacylglycerol, which is the sort of precursor to a phospholipid. Then you actually add the polar head group to it, and the lipid becomes part of the outer leaflet, or the cytosolic leaflet, of the endoplasmic reticulum. But here's the problem. If you're only adding lipids to one leaflet of your membrane, what's going to happen over time? So you just keep adding more and more phospholipid to one monolayer of your bilayer. What's going to happen to your bilayer? What's going to happen? Yeah, one side is going to be longer than the other. And what's that going to make the bilayer do? Well, maybe, but before it does that, she just did it. Do that again with your hand. Yeah, it's going to go like this. It's going to curve. All right? And, I mean, a little curvature can be a good thing, but too much curvature can be a bad thing. And so the cell needs to do something about that. It needs to redistribute those lipids. And it does that using a type of protein called a scramblase. And this is a three-dimensional structure of a scramblase. You can see a whole lot of secondary structure in here. Which secondary structure is that? Alpha helix. Oh, yeah, a lot of alpha helix. And the two lines here represent the two planes of the monolayers in the ER membrane. And so what the scramblase does is it reassorts lipids between the inner and the outer leaflet. So it equalizes them. So that takes care of our curvature problem. Scramblases are interesting enzymes. Doing something like moving a phospholipid, an amphipathic molecule, across a membrane would seem to be something that requires a lot of energy. But in fact, scramblases are ATP independent. They don't need to hydrolyze ATP, and they don't use any other high-energy source. What they do is they form this huge complex that essentially has a hole at the middle of it. And so what they do is they fix it so that the phospholipids don't have to go through the nonpolar environment, the interior of the membrane. They never see it. They pass through this pore, if you will, at the center of this big complex. So scramblase is then going to reassort your phospholipids here so that now you have a more or less random or uniform composition in the two sides of the bilayer. And everybody's happy, right? But the answer is wrong. Because if we look at actual cell membranes, if we take apart cells and we look at who's in the membrane, and specifically we look at who's in this half of the membrane and who's in that half of the membrane, we find out that just like our lateral diffusion, we don't have a random assortment of phospholipids in the two halves of our bilayer. In fact, if you look at the plasma membrane of a cell, the exterior side of it, the side that is facing away from the cytosol, contains almost exclusively two kinds of phospholipids: phosphatidylcholine, which is the most common one, and then that phosphorylated sphingolipid, sphingomyelin. So those are represented here by red and by brown. And so in the Golgi, in the plasma membrane, you find pretty much just sphingomyelin and phosphatidylcholine in the outer or exterior leaflet of the membrane. On the other leaflet of the membrane, you find a very different set of phospholipids. You find primarily phosphatidylserine in green and phosphatidylethynolamine in yellow. And so these membranes of the Golgi and the plasma membrane, very polarized lipid compositions. You know, how did it get that way? I mean, remember, you were adding lipids in the smooth ER, and then the scramblase was reassorting them. So what happened to give you this asymmetric distribution? Well, what happened was the activities of a couple of types of proteins called flipases and flopases. These are enzymes. Anything you hear with an ace on the end of it is an enzyme. And these, unlike the scramblase, are ATP-dependent enzymes. What flipases do is they take phosphatidylserine and phosphatidylethynolamine that they find in the non-cytosolic side of the membrane, and they move it over to the cytosolic side. And they use energy from ATP to do that. Flopases, which were discovered later, do the opposite. They find phosphatidylcholine and sphingomyelin in the inner leaflet here, the side facing the cytosol, and they move it to the outer leaflet. So phosphatidylserine, phosphatidylethynolamine going toward the cytosol, phosphatidylcholine, sphingomyelin going toward the non-cytosolic side or the exterior. If we're talking about the plasma membrane, that's the exterior of the cell. What about if we're talking the Golgi or another organelle? Which is the non-cytosolic side? The outside or the inside? The inside. It's the inside. So if we're talking about the Golgi, it's the lumen, it's the center. And so flipases and flopases are giving us this asymmetric distribution of phospholipids. And interestingly, and you can hopefully be reading your extra credit sheets there, in cancer cells, that normal distribution is thrown all willy-nilly. You start to find lots of phosphatidylserine and phosphatidylethynolamine in the outside layer of the bilayer. And people are still trying to figure out exactly why that happens, and then more importantly, is there any way we can use that as a therapeutic target? So we talked about the topology of membranes inside the cell. And when membranes move around the interior of a cell, they do so in the form primarily of closed vesicles. When you make a vesicle from a closed compartment like the Golgi, you actually get the membrane to bulge out and then pinch off a little bubble of membrane. And here we've tracked the inner leaflet, the non-cytosolic leaflet, and the cytosolic one, the exterior one, if you will, in two different colors. Inner one is in red, outer one is in green. When you form a vesicle, you maintain that same topology. That is, what was facing the cytosol is still facing the cytosol. What was facing the interior is still facing the interior. When proteins are part of this membrane, they can become modified. And we'll look at this later, but when proteins are part of the membrane of the Golgi and the endoplasmic reticulum, they can have sugars added to them. And the enzymes that do the adding are found in the lumen, in the central cavity of those organelles, the ER and the Golgi. So the sugars only get added to this side of the protein. The part that's sticking out into the cytosol never sees those enzymes, so it doesn't have any sugars added to it. So when a vesicle buds off, the sugared part, the glycosylated part, is facing the lumen of the vesicle. But when this vesicle fuses with the plasma membrane, now things turn inside out. So imagine this thing coming up here and contacting cytosolic face to cytosolic face, and then the membranes are going to fuse, and this interior face is going to fuse with this non-cytosolic face, and what was inside the Golgi or the ER is now outside the cell. Think back to that diagram of the membrane where we noticed that all the sugars were on one side. Do you understand now why they're all on one side? Okay? They were all put on to the non-cytosolic side of proteins in the ER and in the Golgi. And then when the vesicles carrying those proteins fuse with the plasma membrane, what was inside the Golgi is now outside the cell. And so that's why the sugars on the glycolipids and on the glycoproteins all end up on the extracellular face of the plasma membrane. Now, Golgi vesicles don't just fuse with the plasma membrane. They can fuse with organelles like lysosomes, endosomes, and they can go back to the endoplasmic reticulum and fuse there. Where's that non-cytosolic side then? Is it facing the cytosol? Couldn't do that without turning the vesicle inside out. You maintain the topology. So when the vesicle fuses, think about it going back where it came from. What's inside, still inside. It only becomes outside when you fuse with the plasma membrane. Okay, there are lots of different kinds of membrane proteins, but four big groups that you need to know are shown here: the transporters and channels, anchoring proteins, also known as adhesion proteins, receptors, and enzymes. Transporters and channels, which is going to be our next big topic, are proteins that allow solutes, specifically polar solutes, to cross a membrane, which they ordinarily wouldn't be able to cross on their own. They couldn't get across a lipid bilayer, but with the help of a protein they can do that. Anchors or adhesion proteins link cells to things that are outside. Those can be components of the extracellular matrix, the non-cellular part of the environment that surrounds a cell, or they can be proteins on other cells. Cells are linked to other cells. That's how we form tissues. That's how tissues form organs. Receptors are ligand binding proteins. They are membrane proteins that have sites that allow them to specifically interact with some particular small molecule. And when that molecule interacts with the receptor, it causes something to happen. So that inside the cell, there's a system for understanding, for being aware that something has bound to the outside of a cell. And finally, membrane-bound enzymes are just like any other enzyme. They are able to catalyze some reaction, principally here in the cytosol, that will convert one compound into another one. Only difference between that enzyme and a soluble one is that these guys are stuck in the membrane. And there are lots of different ways that proteins can interact with the membrane. We just saw the scramblase with all those alpha helices going through the membrane, right? And when we looked at alpha helices and beta sheets, we talked about something called a beta barrel. Anybody remember the beta barrel? It was that tubular structure made out of a folded sheet of beta helix. That can penetrate a membrane. So here's a beta barrel here. Alpha helical membrane proteins can cross the membrane once, or they can cross it multiple times. So here we have a protein that started here, it went across the membrane, then it came back, then it came back. You can have alpha helical domains that span a membrane 3, 5, 7, or more times. Some proteins are only associated with one of the monolayers of the lipid bilayer, and it's usually, again, through an alpha helix here. And so it's kind of like it's turned sideways, and it's stuck in there into the hydrophobic or nonpolar interior of the membrane, but only on one side. It doesn't penetrate all the way through. Other proteins are not going to penetrate the membrane, but they are going to be lipoproteins. They're going to be covalent conjugates of lipid molecules to protein molecules. And the lipid part of the lipoprotein can actually insert into the lipid bilayer and live just happily in there, and once it's stuck in there, it doesn't want to come out again. There are a number of different kinds of lipid-anchored proteins. One type is shown here. Another is shown here. It's actually an integral membrane protein that has an alpha helical domain going through, but then part of the cytosolic portion of the polypeptide has a lipid anchor attached to it that joins it to the membrane. Then finally, you can have peripheral proteins, proteins that aren't inserted into the membrane in any way, but they interact either with integral membrane proteins, as shown here, or they interact with the lipids. Now, these things are loosely associated with the membrane. The original definition of a peripheral protein is you could wash it off a membrane by just putting some salty buffer onto the cells. And what that illustrates is that these interactions are non-covalent. Non-covalent interactions can be easily disrupted by simply changing the chemical conditions. The cell cortex is part of the cytoskeleton. The cortex means anatomically the outer layer of something. And so the cell cortex is the part of the cytoskeleton that's right underneath the plasma membrane. And what we've done here is we've peeled off a piece of plasma membrane and flipped it over so that you can see these cytoskeletal filaments attached to the cytosolic leaflet of the membrane here. And these tubular structures that you see represent transmembrane proteins. They don't actually have to be channels or pores like this. They could just be alpha helical proteins that are stuck through here. And there are actually interactions between the cytoskeleton and the cytosolic tails of many of these membrane proteins. Some of them are direct. Some of them are indirect through so-called attachment or adapter proteins that serve as intermediaries between the cytoskeleton here and the membrane proteins up here. Why does this matter? Well, from a structural point of view, this kind of an arrangement helps to strengthen the plasma membrane, makes it less fragile, allows the cell to be poked and prodded and have stress applied to it by its surrounding environment without ripping apart. So you can just think of it as kind of like the underpinning of the membrane. And that's certainly true, but there's more to it than that. These cytoskeletal attachment points also help to localize proteins in the membrane. Remember that immunofluorescence picture I showed you where you saw all different kinds of proteins aggregated into places rather than randomly distributed. One of the explanations for that, and it's not the only one, but one of them is that certain proteins are localized. They are kept in one particular spot in the membrane by virtue of their interactions on the cytosolic side with this cell cortex, with elements of this peripheral cytoskeleton, which include protein spectrin, the filamentous protein actin, and certain other elements of the cytoskeleton. And so not only is this cell cortex helping to provide structural stability to the membrane, it's also functioning as an anchoring point for certain membrane proteins. And that's one way that you can tether a protein or keep it from diffusing laterally. But there's some others. Instead of tethering to the cytoskeleton inside the cell, you could be tethered to the extracellular matrix outside the cell. An extracellular matrix is made up of a mixture of different proteins and polysaccharides. Collagens would be one example. Fibronectins would be another. There are a number of different components to the cytoskeleton, or excuse me, the extracellular matrix. And incidentally, those proteins are made by cells and are then secreted into their surrounding environment where they assemble. So some of your membrane proteins will be attached to that. Others will be attached to proteins on other cells. And that's illustrated here in an epithelial layer. That is a tissue structure in which you have cells joined tightly to one another to form a sheet. And you find epithelial layers throughout the body. You find them in your skin, that's the most obvious one, but you also find them in the linings of all of your organs. You find them in some of the interstitial structures between organs in the body. And in these epithelial layers, individual cells are interacting with their neighbors through protein-protein coupling here to form junctions. And there are different kinds of junctions that you'll talk about later. There are tight junctions, there are desmosomes, there are plasmodesmata in plants. There are all kinds of different connections between cells. And all of them involve non-covalent interactions between membrane proteins in one cell and those in another cell. Some membrane proteins are tasked with anchoring epithelial layers to what's called the basal lamina. And the basal lamina is just a sheet of extracellular matrix material that these cells themselves secreted, and basically it created a foundation on which they could build this tissue structure. And the cells are linked to that by protein-protein interactions, extracellular matrix protein here, integral membrane proteins here. Then, of course, there's this idea of lipid rafts. Are some proteins kept where they are because they are aggregated in this structure that involves certain phospholipids, cholesterol, and certain other types of proteins? And those lipid rafts, could they be anchored to the cell cortex? Active area of research, lots of people who say yay, lots of people who say, not quite so sure. Some people who like to be contrarian say you don't have any evidence for that at all. And you know, the problem is that it's still an open question right now. And so you'll see in some textbooks, you'll see online, you know, this is an established fact. This happens, blah, blah, blah. But if you go into the literature, it's like, not quite so fast, Johnny. There's a lot we still don't know about this. And it's even possible that lipid rafts could be an artifact altogether. Now, if you really want to piss off membrane biologists, go in there and tell them lipid rafts aren't real, and they'll really blow their stack. But again, there's a lot that we still don't know about this, and so only time is going to tell whether this becomes permanently incorporated into our idea of how cells function, or whether it goes the way of some other popular ideas that just didn't end up being supported by the evidence in the end. Cells are surrounded by a carbohydrate layer. It's called a glycocalyx, particularly if you're an animal cell, eukaryotic animal cell. They have a real extensive glycocalyx. And this is just made up of the glycoproteins and the glycolipids that were formed way back when in the endoplasmic reticulum and in the Golgi apparatus by the attachment of sugars to lipids and proteins in the lumens of those organelles. And then when vesicles budded off, those sugar residues were still attached to the proteins and lipids, still on the lumen side, but when the vesicle fused with the plasma membrane, that lumen side was now exposed to the exterior of the cell. And so that's why all these carbohydrates are out here. And they're not just there for decoration. It turns out that they mean something. Now, one of the things that the glycocalyx does is provide actual physical protection. There's a lot of water that binds to sugars, okay? And you form almost this jelly-like layer around the cell. And that creates a mechanical barrier that helps to protect cells from damage. So damage protection, one big part of what the glycocalyx does. Lubrication, enabling a cell to move. Having a slime layer on your surface is quite helpful if you actually want to crawl around or slip past another cell while a tissue is under construction. But there's more to it than that. Cell-cell recognition. We've talked before about protein-ligand interactions. The ligands for many proteins are carbohydrates. They're these sugars that are attached to lipids and proteins on the surface of the cells. And so when another protein needs to interact with these proteins, they're not just seeing the protein part, they're seeing the sugar part too. And they have to see the right sugars or they won't interact. Finally, cell adhesion, which is just another form of protein-ligand interaction, very often involves contacts between ligand binding sites on one protein and carbohydrates that are attached to lipids and proteins on another cell. And you'll very often hear about the antigens on human cells, for instance, trying to identify cancer cells having unique antigens. Those can be proteins, but more often they're glycoproteins. And the sugars are an inherent part of that antigen structure and an inherent part of what makes one antigen different from another one. And these sugars, again, all go back to the Golgi apparatus and the endoplasmic reticulum, where they were first assembled onto protein molecules that are inserted in the membrane. Good example of interaction between proteins and carbohydrates is the way that white blood cells called neutrophils recognize a place in a blood vessel where they ought to stop during their journey with the flow and actually exit the blood vessel, get out here into the surrounding space. So these are immune defense cells. They're sometimes referred to as the first responders of our immune system because they're usually the first cells to arrive at the site of an injury or the presence of a contaminating foreign organism. And these things are picking up chemical cues from receptors in their cell membranes. When they do, they will upregulate specific oligosaccharide-coated protein molecules that you find in their surface that can bind to proteins called lectins that are attached to the cells lining the blood vessel. When they bind to those lectins, they will undergo a physical change. They will actually squat down, spread out along the walls of the blood vessel, and eventually squeeze through the space between individual cells that make up a capillary into the surrounding tissue and then migrate to the site of infection. We'll actually watch a little movie of this here. So here we've got some white blood cells cruising down through a capillary, and now there are diffusible chemical cytokines that are binding to receptors here on the endothelial cells that line the blood vessel. They're going to upregulate these molecules called selectins that bind to the carbohydrates on these leukocytes, allowing them to attach. So they are actually now going to attach to the surface of the lining of the blood vessel here. When they become attached, they're going to rearrange their cytoskeletons so they begin to flatten out, to spread out along the lining of the blood vessel. And they will actually squeeze through the spaces in between cells into the surrounding tissue here and get at the source of the infection. So here we have a rolling leukocyte along the interior of a blood vessel. This is in a mouse, by the way, and you can see the flow of the blood here. Here's another rolling leukocyte here. Now he's found the junction between capillaries here. Here's a guy who's already squeezing through. This one here is beginning to now. And you see several out here that have already migrated into the extravascular tissue here. And ultimately, this is all about the ability of certain proteins to recognize certain carbohydrates that are attached to lipids and proteins on cells, and to then use the fact that they have attached as a signal of sorts to cause the cell to change what it's doing, to rearrange its internal structure, and to then actually respond in some particular way, in this case by moving, by crawling through the space in between. And you notice here when you got the blood flowing full bore here, these cells are still rather tightly attached here as compared to somebody that's just bouncing along with the breeze here. So the binding of proteins to the carbohydrates of glycoproteins and glycolipids is key to the ability of cells to recognize other things, to recognize other cells, to recognize other protein structures. So here are the things you want to know about cell membranes, their locations and what they do in eukaryotic and prokaryotic cells. What's the basic structure of a membrane, that lipid bilayer, plus the embedded proteins. Why do we have lipid bilayers? All about the hydrogen bonding of water. What are the major groups of lipids that we find in cell membranes? How do lipids move within a bilayer? Diffusing laterally, rotating, flexing, but not flip-flopping very often unless they have help from a specific type of protein. How do sterols affect the fluidity of a bilayer? Do we find them only in animals? How do we know that proteins can also diffuse as well as lipids? Remember the Fry and Edison experiment. We know that membranes aren't homogeneous. We know this because of fluorescent antibody studies. What is this lipid raft concept, and could that explain maybe the non-homogeneity of membranes that we see? Where do you make phospholipids? Smooth ER. Remember, it's the side facing the cytosol, not the lumen. There's a tendency for people with organelles to think everything happens in the lumen, not making phospholipids. Know about the topology of vesicle fusion with other membranes, including the plasma membrane. Be able to describe what scramblases, flipases, and flopases do, and who needs ATP and who doesn't. How do you distribute these four major phospholipids in membranes? What are the four major classes of membrane proteins that we talked about? The ways they can associate with lipids. What is the cell cortex? What are its functions? And blah, blah, blah. So that gets us through the structure of membranes. Next time we're going to start talking about how membranes engage in transport, how things move across them. Any questions before you go? All right. Everybody have a good day.
Make sure you turn in your extra credits.
Yes, bring your extra credits down here because we will not accept them outside of class. You can't like come tomorrow and say, oh, I forgot to turn it in. Got to turn it in now. Yeah.
Did you say that the Golgi line is that considered the outside?
Yeah, if you think about it. So here's a Golgi, right? There's the lumen there. Now we're going to make a vesicle from it. That means we're going to bud off part of the membrane and we're going to close it up again. So inside of the vesicle is still just like the inside of the Golgi. But now this thing goes up and it meets the plasma membrane, and the membrane fuse, it opens up. So the side away from the cytosol is always the side away. Rather than worry about what's the outside of the cell, got a little animation. Okay, it means that it can't do what it does without hydrolyzing. I don't know the mechanism. All it's just hypothetical.
Would it reduce?
It's a like I said, don't overthink it.